Hi everyone, welcome to this episode. This is all about genetic engineering. Have you ever wondered about the foods that we are eating? Just like the image that I'm presenting, the first image that you can see on the screen, there's a white colored rice and there is a yellow colored rice. The second image is the insulin, which is widely used of those individuals suffering from diabetes. The third image is a comparison of um, both or the same age salmon. So you can see there is a difference with regards to their size, their length, and also their weight. And last but not the least is the production of the COVID-19 vaccine. Any guess of what they are a product of? You guys are all correct. They are product of genetic engineering. In this session, I'm going to present to you about genetic engineering. And this is how I structure the video. The first one is to understand the genes or the DNA. The second is to describe genetic engineering. The third one is to identify the processes involved in genetic engineering, present examples of genetic engineering, and lastly, discuss the advantages and disadvantages of genetic engineering. So just relax, just chill, and let me present to you about this topic, genetic engineering. So the first question is, what are genes? Genes are the basic physical and functional unit of heredity. Genes are made up of DNA, or the deoxyribonucleic acid. Some genes act as instruction to make molecules called protein, and this protein will give the phenotype of our features or ourselves as an individual or as a species. However, many genes do not code for proteins. In humans, genes vary in size from a few hundred DNA bases to more than 2 million bases. You will see from the image or from the figure that I'm presenting right now that in here you will see that this is the cell, and inside the cell there is nucleus, and inside the nucleus it is made up of millions and billions of this DNA. Translated, or this DNA are created or formed by chromosomes. And these chromosomes are made up of the blueprint of our genes, which is known as our DNA, or the deoxyribonucleic acid. Looking back, or if you're going to zoom this DNA, you will see this kind of structure. So to make it like really quick, there are 4 bases of our DNA. So we have thymine, we have the adenine, cytosine, and also the guanine. In DNA, A is always paired with T, or vice versa, and C is paired with G, or vice versa. Once again, A is paired with T, G is paired with C, or vice versa. And there are sugar phosphate backbone. Those are the base pair, which is the nucleotides. And there is also a 5-carbon sugar that is present here, which bind via polypeptide bonds. And these base pairs are paired with a hydrogen bonds. So what is exactly this genetic engineering, and what is the importance of that? So genetic engineering is the changing the genetic material of an organism by removing, modifying, or inserting individual genes. Genetic engineering is a lot like selective breeding, but a much faster process by taking favorable genes from one organism and implanting it into another organisms, known as the DNA. This can be done in a laboratory with special equipment. So, from my first slide, you will notice that I presented four examples of this genetic engineering. Number one is the golden rice. The second is um, the insulin, which is laboratory prepared, coming from the bacteria. The third one is the GMOs, or the genetically modified organism, which is the salmon. And the fourth one is how to create or creation or making or formulating a vaccine made up of genetic engineering. So in this video, I will also explain to you what are the different processes involved in this genetic engineering. So here we go. Let's begin with the first. The first one is to obtain the human chromosome containing the insulin gene. So you will notice in here, this is the insulin gene, which is included or a part of the pancreas, because the pancreas is the one which will create this insulin. So the pancreatic nucleus, it has this insulin gene inside the nucleus, of course. And then after that, this insulin gene of the human will be cut using restriction enzymes. So you will see, or you will notice in here, that it forms this sticky end or sticky ends both sides, and that is being cut by a restrictive or a restriction enzyme. So what will happen afterwards? After cutting and making the sticky ends of this insulin gene coming from the human pancreas, there will be base that can be paired with a complementary basis to form a double strand. So you will see in here, it has been cut, and it forms this fragment of DNA containing this insulin gene. So this process is what we call isolation. We are isolating the human gene and cut it using the restrictive enzyme. Next, after that, is to obtain the plasmid from the bacterium. Looking at the general structure of the bacteria, it has this plasmid, which is circular in nature. And so we have to isolate them also, just like what we have done in the human gene, which is the human insulin gene. And then after that, we have to cut this plasmid using the same restriction enzyme that we use in cutting the insulin gene from the human, or from the human gene. And this one will gonna produce this sticky ends too. Same thing with the insulin gene that we have cut before. After that, this needs to be mixed, or they have to be combined. So mix the plasmid with the DNA fragment containing the insulin gene. Add the enzyme DNA ligase, or we have to use DNA ligase in order, or it's an enzyme, in order for us to bind or to join the insulin gene and also the plasmid, which is being cut. And so after that, we will have this product. This product is known as recombinant DNA. So after that, this recombinant DNA, it has to put it back to the body of this prokaryote, which is the bacteria. So in order for us to put it back to this recombinant DNA, there should have an application of a temporary heat, so or an electric shock, so that uh, the cell surface membrane of this bacteria will be able to open, or it will create a pores, and this pores, it will enable this recombinant DNA to be able to get inside this bacteria. And so after that, we can call this as transgenic bacterium. The process is not yet done. The process is not yet completely done, because this bacteria, it needs to replicate, it needs to duplicate in order for them to become, to reproduce its number. So, next to it, this transgenic bacterium or bacteria, they need to be placed in a vat or in a fermenter, supplied with essential amount of nutrients for them, or glucose, that will be used in order for them to be, as part of their nutrition. And as they respire, more and more insulin, they will produce more insulin for that is safe for human consumption. Now, so that would be right. So next is what are the examples of genetic engineering? So from my explanation in the steps of genetic engineering, the very first example of genetic engineering is the creation of the human insulin out of or from the bacteria, specifically the E. coli. So people have taken the insulin making gene from humans and inserted it into bacteria, causing the bacteria to make human insulin. So why the scientists have to do that one? The bacteria can reproduce quickly, causing them to produce insulin at a much faster rate. Yes, we know that one, that bacteria or bacterium, they are able to reproduce in a split of a second. Or in every 28 seconds, there are new bacteria that will be able to be produced or to reproduce via binary fission. That's a process. You will see from this video it's like the same thing from the steps on how genetic engineering works. So isolating the human insulin gene, there are a section of the human DNA, and part of it, this human DNA, we have this human insulin gene. And so this human insulin gene will be cut using restriction enzyme. And this restriction enzyme, it will be used, this restriction enzyme will be used in order for the bacterial plasmid to be cut too, making or forming sticky ends on both sides. So human insulin should have a sticky end side, and also the bacterial plasmid should have a sticky end, and they will be complementary to one another. After that, after cutting, this genes or this fragments of the human insulin gene will be able to be joined by means of the ligase enzyme. So the ligase enzyme will be used in order for them to be to join together, and it will form this recombinant DNA. And after that, this recombinant DNA, which is basically the plasmid, an edited plasmid, it will be returned back into the body of the bacterium. So, by applying electricity or heat, their pores will be open, specifically their cell walls, and then after that, this recombinant DNA will be able to get or to be inserted inside. And then this bacteria, they need to replicate or duplicate to reproduce with the same kind, having a recombinant DNA with them, or recombinant plasmid within them, and so they can grow in the fermenter. And then in the fermenter, as they respire, their product is the insulin, which will be separated and also purified in order for this insulin to be safely consumed or to be injected by the diabetic person. This is the same thing in here. So how could we make the bacteria to become transgenic? It's the same explanation from before. So we have at least 6 steps in here. So additional information, the plasmid or the recombinant DNA, all right, which is the circular DNA of this bacteria, they act as a vector because they are vector because we put the human insulin in there, or the human insulin gene into the bacterium in order for them to produce this insulin. Aside from that, this will be the summary version of how the bacteria can be manipulated to produce human protein such as the insulin. So we have a total of 5 procedure or processes in here. I highlighted the terms that is very essential in this study. Number 1 is restriction endonuclease enzyme or restrictive enzyme. The second one is the enzyme ligase, which will form a recombinant plasmid or recombinant DNA. And then we have to allow the bacteria to reproduce, making more cells which produce the protein. So the protein in here will be the hormone insulin. Second example of genetic engineering. So people have taken bad genes or genes which cause cancer and other disease out of a person and inserted healthy genes into a person to cure them of disease. But however, they may be cured of that disease, but they may be able to get another worse disease because of being implanted with other genes. This process or this particular examples of genetic engineering is what we call stem cell cure or stem cell injection, which is could be used also for cosmetic surgeries or cosmetic purposes. The third one is people or scientists have taken a weed-resistant gene and inserted the gene into a certain crop plants. The example of the gene which will be inserted into a crop plants, it will be the Bt genes. So that Bt genes, it has a resistant to the weed or to the herbicides. So farmers can now spray weed killer on the crops to kill the weeds, but the spray will not affect the crops. And so the crops will grow better and bigger because weeds do not take away sunlight, nutrients, and water. But there is a disadvantage of this. This gene could leak over into the weed population, and sometimes it could cause the weeds to be resistant to the herbicide too. So this weed-resistant gene, this is not that effective in killing or in eliminating the weeds, which could cause or which could harness them to be able to grow and also to increase their yields, right? So you will see from the image in here that the BT or the Bacillus thuringiensis, this is a kind of bacteria wherein that gene is included in them. And so this BT gene, it will be inserted into a crop plants, all right, in their before the fertilization process occur. And so if they will grow, if the seeds grow, um, and grown into a plant, therefore the pest will not be able to feed on them and they will be killed. So the effect of this one, of this gene inserted into the crops, is that there will be more yields, especially for the corn, and say for um, consumption. Next is that humans have injected genes into the crops to make them grow in colder environments. And humans could have injected genes into crops to make them grow in dry or drought environments. So you can see in here from the image below that even if there is an extreme weather condition, both extreme weather condition, extremely cold and extremely hot, both of these organisms or these plants, species of plants, will be able to survive. And genetically engineered fish, which is specifically the salmon. So the genes have been inserted into a fish to make them grow twice as big and twice as fast. You will see, or you will notice in here, that both fish are 18 months old, and so there will be a changes with regards to their length and also their weight. So the length is 24 inches for the GM or the genetically modified salmon, and it has 6.6 pounds in compare with 2.8 pounds for the farm salmon. So literally, if that's the case, if we are consuming this GM salmon, I mean, this is very useful for those who are farming them or growing them, because economically wise, they could produce a lot of profit. Genetically engineered chickens. Genes has been inserted into chickens, specifically the embryo, to make them grow twice as big and twice as fast. They are also big to the point that they can't walk. So same thing with the GMO fish, that they can grow as big as they can, and so when they are sold into the market, or when they are put into the market, therefore they will be able to weigh more. It means that more profit. And last but not the least is this one is a kind of an interesting subject, which is all about how to develop a virus vaccine. So right at this moment, the world is experiencing pandemic, and in order for us to have this herd immunity, we should have this vaccine. So the question is, how safe it is? What about the efficacy? So clinical trials already have shown the effect or the impact of this vaccine to the humans. All right, so let's try to figure it out how this virus vaccine has been formed and created. The first thing in here is that there should have an isolation process. The isolation process in here is that they have to create, scientists or geneticists or microbiologists have to create a chemical serum. And that chemical serum, it has to include, or it has to contain this virus. This virus can be weak or attenuated. This could be a killed virus or a dead virus, which is inactivated. Or we can use, or the scientists could use, an isolated pieces of the virus subunits, specifically its antigens. Antigens are special proteins that can be found at the surface of the pathogen. But in this case, this is a virus. So this virus has this protein which is located at the surface of it. And so, aside from that, it could also put some genetically engineered particular virus genes and capture their blueprint. It could be coming from their DNA plasmid from the inside of them, inserted into the plasmid of the bacteria. We can also embed a blueprint of the RNA, which is there at the middle of it, and also embed blueprint in an adeno virus. Now, this these samples that came from the isolated virus, this will be put, these particles will be put in a solution, and then it will be tested first into the rats or into the subject. So they have to be tested first in an animal or in the animals. And then if there will be some responses, if it prompts an immune response, therefore, it could be tested into humans. So, but if there's no immune response to the animals, therefore it has to stop testing and go back once again to the first step or the first processes. This process involves an accurate and also meticulously laboratory prepared equipment. So let me go back in here. So if it is not toxic, or if it shows some immune response to the body of the animal, therefore this vaccine is safe. So it will be tested first into the sample, which is 10 to 100 person, or from 10 to 100 people. Now, if the testing that has been done is good, therefore it can go to the next process or procedure wherein this will be tested into more than 100 people. And so, if there's no failure, if the efficacy is good, it will be continued up to 10,000 people and more. It will be tested in there. And so, if the efficacy is quite good, and if the immune response is high, therefore the efficacy is high, it will create memory cells. Therefore, the procedures that has been used in here, it will enter a mass production. And in that mass production, therefore, we will be able to create this medicine or this vaccine that is actually made up of genetic engineering. The process is quite complicated, and it's time consuming too. That's why, as you know, this one covers like a year before the vaccine has been produced and created. But good thing, at the end of the tunnel, at the end of the dark tunnel, there is a light, and we are near to end. Hopefully, we can end this pandemic soon by creating or by herd immunity. So that's the promise of the genetic engineering process. Last part of my video is about the advantages and disadvantages of genetic engineering. So you will see in here that genetic engineering may work wonders, but it is all a process of manipulating the natural. Once again, genetic engineering is quite the same process of selective breeding, wherein we choose the phenotype or the specific characteristics that we would like to have, especially for the plants and also for the animals. So here are the advantages and the advantages of genetic engineering. You can see that um, the positive sides could have a better taste, nutrition, and growth rate. We could produce pest-resistant crops and crops with longer shelf life, especially the corn from, the sweet corn that we are enjoying currently in the market. Humans have been able to produce new foods which couldn't be reproduced in nature, and humans can help cure certain disease like cancers. And right at this moment, the clinical trials for the HIV cure is ongoing. So that's another breakthrough that is caused by genetic engineering. And of course, it boosts positive traits and suppress negative ones. Talking about disadvantages of this genetic engineering, though we can create higher yield, uh, uh, good tasting crops, better appearance crops, but some of them could lead into allergies. And aside from that, it could create accidentally made some crops which have less nutritional value because just like I mentioned to you earlier, since we modify the plants or the crops or the animals, it could lead also to some less nutritional value. And how about the accidental gene transfer? Such as when we inserted genes into crops to make them resistant to pests or herbicides. The genes leaked over the weeds and made them resistant to pests and herbicides too. So the purpose is not actually like it's not effective in if that's the case, since the genes can leak, and so pests, I mean the weeds can absorb these genes that is special only for the crops. Therefore, later on, these weeds, as they grow and produce their offspring has this ability to be able to become resistant to pests and also to the herbicides too. And genetic engineering in human beings can have certain side effects. So while treating one defect or disease, another one could lead into the worst. And what's next? How about inserting special genes into the babies to have the children that are smarter, muscular, prettier, certain color eyes, certain color hair, and talented? Do we play God? Now, I would like you to join the discussion by scanning the QR code that I prepared here. I'm giving you a minute to scan it and to go through with this Padlet. You may pause the video for a while in order for you to have a better view so that you can scan this QR code. And lastly, this is the references that I use in my presentation. There you go.